Discovering green turtles' foraging habitat in the Galapagos Islands using stable isotope analysis by Sabrina Bocek, Unity Environmental University, Master's Capstone Project, with special acknowledgement to project partner Garrett Lemons, NOAA Fisheries Biologist, Marine Turtle Ecologist, and Stable Isotope Lab at Southwest Fisheries Science Center. Stable isotope analysis can help us trace key elements through the food web into the metabolized tissues of the individual, giving us a longer observation of the habitats it forages in, especially important for monitoring migratory species and endangered species. The stable isotopes used in our study were of carbon and nitrogen. An isotope is the same element, but with a different number of neutrons, giving it a different mass. In our study, we did carbon 13 and nitrogen 15. The stable isotope signatures of the individual's tissues tell us what trophic level they consume from. This is from the nitrogen 15 stable isotope, which has a gradient with a 3 to 4 parts per thousand stepwise enrichment for each trophic level. So the bottom would be herbivorous, and then omnivorous, and then carnivorous. And there's a distinct difference between terrestrial isotopic signatures and marine ecosystems. Carbon 13 on the bottom axis, when it is less negative, actually means that it's more productive, and it reveals the location of the plants that are consumed. The lower the number, carbon 13, means that we are benthic and closer to shore, and can also predispose that the individual resides in that habitat. Less productive and lower amounts of carbon 13 reveals food sources found in the pelagic zones. Our species of interest are the green turtles, Chelonia mitis. They are considered sentinel or indicator species, which means their abundance, distribution, and health in the ecosystem are reflective of environmental conditions. They have an estimated life expectancy of 70 years and reach reproductive maturity around 25 to 35 years of age. The green turtles were listed under the Endangered Species Act in 1978, and one of the major recovery actions for conservation includes protecting their foraging habitats. NOAA Fisheries is currently working to designate those hotspots for feeding grounds. The green turtle is a circumglobal species primarily found in subtropical and temperate regions of the Atlantic, Pacific, and Indian Oceans. Sea turtles are migratory, traveling thousands of miles between natal beaches and foraging habitats. It is vital to locate their foraging habitats and the degree of site fidelity, along with their primary diet items. Our study collected 45 tissue samples between Fernandina Island and Isabella Island in the Galapagos. The Galapagos consists of 19 islands 1,000 kilometers away from the South American continent and is the largest marine reserve in the world. The methods consisted of tissue samples of suspected plant and invertebrates within the habitat, along with the collection of 45 skin biopsies of green turtles between the two islands over 3 different seasons. Tissue samples from potential prey species, hereafter referred to as habitat samples, were gathered opportunistically during field line transect surveys in noted areas for each island, as well as opportunistically during fieldwork. Whole organisms were collected for all species except for eelgrass, from which only blades were gathered. Turtles were captured in the neritic zone within large gill nets with proper standards to ensure no harm was inflicted. Skin tissue, stratum corneum, was collected from dorsal surface with either a razor or biopsy punch. Skin and habitat samples were frozen before processing in lab, where after thawing they were dried out and ground into small grains and homogenized into a fine powder using mortar and pestle. To remove lipids, here's the lipid extraction machine. The samples underwent extraction using a Soxhlet apparatus. 1 milligram of green turtle skin or habitat samples were placed in sterilized tin capsules at University of Florida's laboratory. Analysis was conducted using a continuous flow isotope ratio mass spectrometer. When the particles enter, the magnetism separates the isotopes by mass, and then the detector reads it at the other side. Here we have the table of the carbon-13 and nitrogen-15 found in prey items and the total of all the sea turtle average between both sites. Stable isotope signatures of carbon and nitrogen found in turtles' tissue between Fernandina Island and Isabella Island. Before running statistical analysis in RStudio, we need to determine the trophic discrimination factor for the species being used in the mixing model. This factor is the calculation of the food item as it becomes metabolized into the tissues of the consumer. These were predetermined in previous scientific studies on green turtles, giving us the discrimination factors to show metabolized stable isotopes over a 3-month time as it is metabolized into the epidermis. This graph on the right shows stable isotopes found in different tissues in different species and the time period that it reflects. So blood, plasma, blow, breath, and feces are hours to days to weeks. Skin, muscle, blubber, and blood is a longer time period, weeks to months. In our study, epithelial tissue reveals up to 3 months of food metabolized. And so this just shows the study involved that gave us the formula to align metabolism into our statistical models. To elucidate dietary composition, we used a Bayesian mixing model called Stable Isotope Analysis in R, where habitat, which is the food items, and the turtle's skin tissue were modeled with skin-specific correction factors for metabolism. And then we got this, our isospace plot. We can see 2 separate clusters for the carbon-13 axis, revealing 2 separate locations of feeding areas, and could also suggest that some are residents while others are new recruits into the area. And we can also see on the stable isotope of nitrogen 15 axis that invertebrates were above this line and then microalgae was below that line. So our overall population proportion of diet items shows that half the diet was invertebrates, and then the other half of the diet was a mixture of microalgae. In previous studies, different seasons showed that the green turtles in the Galapagos would opportunistically feed on invertebrates during the cold season and then have more of an herbivorous diet in the warm season. Diet characterization directly infers habitat that is critical for foraging, and coupled with other tools such as tagging and telemetry gives the life history changes for foraging habitat shifts. The Galapagos is unique in that it provides both nesting and foraging sites and hosts both temperate and tropical climates. Moving forward, continuous monitoring will help guide management practice and policy to ensure a thriving green turtle population amongst changing climate shifts. And here are the references for this study. Thank you.